Hello, everyone. Welcome to my first ever exam review video on YouTube. This is going to be my Biology 104 Unit 1 exam review. If anyone goes to University of North Carolina, Chapel Hill, or you take a biodiversity class, I hope this video will be helpful for you. I know I made this PowerPoint to help me out with my studying, so that's why I've created it. So let's just go ahead and get into the video and get over some review. Hopefully this will summarize the entire first unit. I've put a lot of studying into this week in specific, so um, I'm hoping to do well on it, and I want to help you guys out as well. All right, so first of all, let's start with some basic definitions. What is biodiversity? Obviously, bio means life, diversity means different. So, biodiversity is the variety and variability of life on Earth or in an environment or in an ecosystem. Functional diversity is the role of each species in that ecosystem. Because, like function, same thing. Genetic diversity is obviously the measure of different genetics or gene pools within a different environment. Obviously, the more gene variation there is in an environment, that's going to make the population more resistant to changes versus if there was just less genetic variation and something happened like a mutation and could really devastate an entire population. Ecosystem diversity, once again, that's a variety of habitats and communities. So if you have an ecosystem that's very diverse, that means there's a lot of organisms, ecosystems, a lot of bugs, critters, however you want to say it. Supporting ecosystem services. There are a bunch of different ecosystem services. My professor in specific wanted me to remember four: supporting, provisioning, regulating, and cultural. Cultural is going to be anything that has to do with humans. So, say a recreation, building something there, having a party there. Just cultural is what people would do in an ecosystem. Regulating has anything to do with what would regulate in an environment from, say, not getting too much of X or not getting too little of X and blah, blah. What would regulate it? So obviously we have climate regulation, food regulation. So that would be like predators, disease regulation. So that would be like I said, different types of immunity and whatnot. So that's everything that has to do with regulating an environment. Provisioning ecosystem services is everything that has to do with humans. So a provisioning service would be like food you can extract from the environment, fuel, water, medicine. That would be what provisioning services would be. And once again, supporting would have to do with anything that supports an ecosystem. What allows an ecosystem to thrive? What allows it to survive? So first of all, we have soil formation, soil nutrient cycling, so the nitrogen cycle and the carbon cycle, and obviously like primary production, photosynthesis, and cellular respiration as well. And just some general review that you would learn in any general biology class, we have our scientific method. We question, we research, we create a hypothesis, we experiment, and then we analyze, and then we go to a conclusion. This is what you've gone over ever since you were in fifth grade. But I mean, if you don't look over it, you might forget it. An independent variable. Once again, every time you do an experiment, you have an independent variable and a dependent variable. The way I like to think of it like this is, well, this is how it is. Your independent variable is the thing that you're changing, and the dependent variable is your measure of the outcome of that change. So your independent variable will be like amount of sunlight that we put on a plant, and then the dependent variable would be how much the plant grew. So the dependent variable completely depends upon the independent variable. The independent variable is just a thing. It just is. The dependent variable is just the measure of that. So independent variable will be like, um, if I go to the gym 30 days a month, that would be the independent variable, going to the gym 30 days a month. And dependent variable would be how much I can bench press by the end of that month. So we could have an experiment. We could have a control group, an experimental group. A control group, that is just the environment that receives no change. The experimental group is the group that receives a treatment. So say in my hypothetical scenario, we have 100 people. We assign 50 to the control group and we assign 50 to the experimental group. The people in the experimental group have to go to the gym every single day for a month, and the people in the control group don't go to the gym every single month, or they live their normal lives. But the people in the experimental group are forcing to go to the gym every day for a month. And the independent variable is 30 days of going to the gym. And the dependent variable will be bench press PRs or personal records at the end of the 30-day period. So our independent variable is the amount of time going to the gym. Our dependent variable is our measured outcome of that. So obviously, some people might be able to do more weight than others, right? Or you could measure it with weight loss, if that makes sense. Independent variable is a variable, and the dependent variable is the measure of that variable. And then we have our control group, which receives no treatment, that we're just watching to make sure what would happen to an ordinary population that doesn't receive a treatment. And our experimental group would be like the group that we test on or that we assign something to. Biological species construct. This is just the idea that if you have two organisms and they are able to like reproduce, then they're the same species. So there would be a question that would say there are two types of organisms that look exactly the same. Are they the same species? But if they can't reproduce and create offspring, then they're not the same species. And a morphological species is just a species that looks visually the same. An endemic species is a species that is only found in one location. So if a species is endemic, that means that it only lives in one area of the globe. So I'm trying to think of endemic species. I wouldn't really know. I mean, are penguins an endemic species? Penguins only live in like Antarctica, I think, right? That's how I remember. I remember the polar bears live in the North Pole and the penguins are in the South Pole, something like that. Whatever. And then eutrophication. This is just another definition. This is the process of, I'll get more into that later with the nitrogen cycle. But basically, this is the process of how if you put too much nitrogen and too much fertilizer on crops, all that excess runoff will get into like water systems, and that will cause like algae blooms, which will basically just absorb all the oxygen, which will make everything in the ocean pretty much just die because they can't get any oxygen. And that's what the name of that process is. But I'll explain more into that when we get into the nitrogen cycle. And this is how this slide here is how we measure like basically biodiversity in an ecosystem. So we can measure it through species composition, species richness, or species evenness. And we could also research through transect and quadrat sampling. So let's say if I had an ecosystem that just had pigs, chicken, and cows, I would say the species composition is pigs, chickens, and cows. It's just the name. So species composition, the name of all of the species in an ecosystem. Species richness would be the number of species in an ecosystem. So for that previous example, the species richness would be 3. So if there was a question that was like, there's an ecosystem, it has pigs, chickens, and cows. What's the species richness? The answer would be 3 because 3 species. And your species evenness would be a measure of the number of species. So you could have a graph of species evenness. So let's say we could have 2 cows, 4 chickens, and 6 pigs. Even though the species richness is the same, the evenness is very, very different. So, or you could have species that have similar evenness. So that's just what it is. That's just a measure of that. Transect sampling. This is just a sampling method. Let's say if I took a straight line and I walked from the beach, I went to the complete east coast of North Carolina and I walked in a straight line all the way to California. And I walked in a straight line and I just took a measure of every piece of, I don't know, every piece of wildlife or every animal. And I took note of that in a straight line. And I used that to calculate species composition of the United States. That would be seen as transect sampling. So just walking in a straight line. And you could have various lines, you could do whatever. Quadrat sampling would be we took small parts of an environment and we just sampled it. So instead of an example of transect sampling would be we'll have 50 people walk in straight lines all across the United States, all at different latitudes, right? Or quadrat sampling would be we'll have 50 people sample each state. So those would be two different methods of sampling, right? So we have one that's surveying a group, or just a circle, and one surveying just walking in a straight line. So those are two different kinds of how you could take measure of wildlife in an environment. And once again, we have, not once again, alpha, beta, and gamma diversity. This is just something that you're going to have to know for this class if you have to understand it. The alpha diversity in an environment is the total amount of species. So in this piece of paper that I wrote down here, I have habitat X and habitat Y. Habitat X has frogs, chickens, pigs, rabbits, and deer. Habitat Y has frogs, chickens, a fox, possums, goats, cows, and squirrels. Okay? So alpha diversity would be the total number of species. So habitat X for alpha diversity would have 5 species because it has frogs, chickens, pigs, rabbits, and deer. Habitat Y would have 7 species because it has the frogs, chickens, a fox, possums, goats, cows, and squirrels, right? So that's alpha diversity. Beta diversity is kind of confusing. You're going to have to really think about me. I didn't understand it at first. You kind of got to think about it. Is when you're comparing two habitats, the beta diversity is the total amount of species between the two groups that the two groups do not share. So if we were trying to calculate the beta diversity for habitat X and habitat Y, we would look at it, we would say, okay, we have the alpha diversity of X and Y is 12, right? So when we calculate the beta diversity, we see that both groups have frogs and have chickens. So it's just exactly what I said. We would take the total amount of species between the two groups that two groups do not have in common. So if you're looking at what species between the two groups that the two groups do not share, it's basically everything else besides frogs and chickens. So we would count pigs, rabbits, deer, a fox, possums, goats, cows, and squirrels. So beta diversity would be 8. And then gamma diversity is just the total amount of species between the two groups, not counting repeats. So like I said, if we combine the alpha diversity of both the groups, we get 12. And then we're not going to count frogs and chickens twice. So if we were just looking at the total amount of species that are present between the two habitats, we would get 10 because we would get frogs, chickens, pigs, rabbits, deer. And then in habitat Y, we're not going to count frogs and chickens. And we would get fox, possums, goats, cows, and squirrels. So that gamma diversity is 10. So I think alpha diversity is pretty straightforward. It's just the amount of species. And beta and gamma diversity, it can be a little bit difficult to comprehend. But I think, from my understanding, it's just a measure of how much an ecosystem can change, say, if something were to happen. So if you knew the beta diversity, you can maybe kind of try to comprehend any displacement issues if there was, say, a hurricane or a bad tornado or whatnot. And this is the Shannon Wiener index. Shannon Wiener index is a measure of species richness and evenness. If you're taking this class, it's just a calculation you're going to need to understand. The Shannon index is just a measurement between the species evenness and the species richness of the two environments, right? Or the environment, right? So this is just going to give us a value of basically biodiversity. And how we're going to calculate this is we're going to take the total amount of organisms total in the entire habitat. So habitat X, if you add the number of frogs and the number of chickens, the number of pigs and the number of rabbits and the number of deer, you get 44. So 10 plus 8 plus 4 plus 20 plus 2 is 44 individuals. And how you would calculate this is you can go in your calculator, any TI-84 calculator, and you do the log of like that number of organisms. So with frogs, we have 10 organisms divided by the total number of organisms, which would be 44. So we would do log of 10 divided by 44, multiply by 3.22. Don't ask me why 3.22. From what I've learned in class, that is just a standard. What's the word I'm looking for? It's a constant. It's just a constant you're going to have to use. So log of number of organisms for X species divided by the total amount of organisms in the environment multiplied by 3.22 multiplied by that same fraction again. Okay? So we would do, for this example of frogs, we would do log of 10 divided by 44 multiplied by the constant 3.22 multiplied again by that fraction of 10 divided by 44. And we would take the sum of that, okay? We would take that, and then we would once again do that for every species, right? So you see here, I did it again. We would do log of 8 divided by 44, log of 4 divided by 44. Once again, you see it for every single species. And we're going to get that, the number for each species, and we're going to add that all together. So in my calculator, I can just literally type it in all at once because I don't have to get every single individual value and just add it up at the end. I can just type it in and my calculator will solve all of it for me. And when I did it, I got negative 1.91. And when you put it in the calculator, it's going to come out as negative, but your Shannon index is always positive. So for the example for that, the correct answer would be 1.91. And when we go over a practice test in a second, I'll verify that math is correct. Rarefaction plots. This is basically just a plot of measuring species in the environment. So, yeah, the species accumulation curve shows the number of species found as sampling effort increases. And when it asymptotes, meaning that when something asymptotes, it becomes straight, right? Like you learn in math, you have what's called a horizontal asymptote, right? As X preaches infinity. That's like calc. But so let's say I sample an environment 1,000 times. And every time I sample it, I keep discovering more and more and more species. So if I was looking at a graph, the X value would be amount of times sampled, Y value would be species found. So if that number keeps going up like linear, that means that it's a very diverse environment because every time I'm sampling and I'm increasing my sampling effort, I keep finding more and more things. But on the other hand, let's say I do 1,000 samples and it like starts off linear and then it like asymptotes into a straight line. So with each additional sample, I'm not finding anything new or anything rare. That gives me a good idea of how many species are in the environment. So this rarefaction plot or plots gives you a good idea of how many organisms are in your environment. And once again, if it's still increasing, that means that you need to keep sampling because you're going to discover more biodiversity and you have a very diverse environment. Or if it asymptotes, that means you already have a good idea of how many species are in your ecosystem. So for this example here, if we were to look at, say, the blue line needs more sampling because it hasn't asymptote yet. It's still kind of increasing. Meanwhile, this purple line at the top has kind of evened out. We're not going to find anything new because it's already a straight line. We haven't discovered any more samples, even though we could have already sampled it, say, 2,000 times. So that's just what that plot is. It gives us a good idea of how many species we've already discovered with sampling efforts. And if we're not finding anything else as we sample more, we don't need to keep sampling. And vice versa, we might need to keep sampling if we keep finding new stuff. All right, GPP, MPP, and respiration. So this is a measure of how much energy is available in an ecosystem available for use. So think about it like this. Gross primary productivity is the total amount of energy produced in an ecosystem. This is just gross. Sorry. So let's say there could be 1,200 joules available in an ecosystem, and respiration might burn 200 of that. So what we would have left would be 1,000. So GPP is the total amount of energy available in an ecosystem, or that's in an ecosystem, and net primary productivity is the energy that's available for use after respiration. And once again, respiration is just the energy released into the atmosphere when plants aspire or respire. So GPP equals NPP plus R, or NPP equals GPP minus R. Just think about it like this. If, say, an environment has 1,200 joules of energy and plants respire 200 of that, then the net amount of energy still available in the environment afterwards is going to be 1,000 joules available for use by other organisms, right? So if you look at these equations here that you probably need to know, if you get a calculation, it might ask you, if you get a question that's the net primary productivity in an environment is, say, 1,000 joules, and plants require 500 joules for respiration, what's the gross primary productivity that was originally available? You know the answer would be 1,500. It's just basic math that you're going to need to understand. Also, if you get another question, plants usually do cellular respiration most at night. Interestingly, I didn't know that until like last week. So if you get a question and it's asking you if you want to measure the R value of a plant, do it at night. You don't do it during the day, because at night is when they're most active, because I guess that's when they're not doing any photosynthesis, from my understanding. But definitely those, if you need to know anything in specific, it's those three equations right there. And I mean, if you understand algebra, those three equations right there are just basically just the same thing, just flipped around. So GPP equals NPP plus R. In that second equation, all I've done is just subtracted R from both sides. So it'd be NPP equals GPP minus R. Yeah, it's very simple if you understand the concept. All right, climate change. So if you understand science, climate change is real. Um, Even though I do love Donald Trump, that's my man. Climate change is real. You look at this graph over here. Since 1974, in a town called Barrow, Alaska, which is actually a town in the very, very northern hemisphere of Alaska, like very, very top, almost close to the North Pole. Over time, we've steadily seen this concentration of carbon dioxide increase in the environment. And as you can see in, say, 2007, it was very, very close to 400. So, I mean, I'm sure it's a lot more than that now. But yeah, carbon dioxide concentration in the environment is increasing due to human uses. And just some over here um, on the right. Why is this happening? Obviously, fossil fuel burning, forest destruction, human use, and just some questions over here to think about is, how does carbon dioxide enter and exit the atmosphere on our plants? It enters through cellular respiration, fossil fuel combustion, fires, forest destruction. Obviously, um, yeah, when humans build stuff, that increases CO2 concentration. And it exits through photosynthesis and is absorbed by oceans. Another thing that's interesting that I learned is a lot of carbon dioxide is absorbed by the ocean. I don't know, that just seems interesting to me. And where are the highest concentrations of CO2 found on our planet? Oh yeah, that would be in a lot of cities, like like Hong Kong, New York. That's has a lot of very, very high concentration of CO2, obviously, because there's a lot of people there, a lot of infrastructure, a lot of factories. So if you get a question of like where would be an area of high CO2 concentration, it would be like big city, a lot of people, highly populated area. And if you had an area that had the lowest CO2 variability, you'd say somewhere like Antarctica. Well, not somewhere like Antarctica, because Antarctica literally has nothing there. There's no people there. There's no factories. There's no plants. There's no trees. So it has the least variability of CO2 concentrations. And another thing is, in what month are CO2 concentrations highest in the northern hemisphere, lowest, and why? So in the winter months, we're actually going to have the highest concentration of CO2 in the environment, because there's a lot more cellular respiration occurring than photosynthesis, and vice versa in the summer. Plants are doing more photosynthesis than they are doing respiration. So if you look at the equation below, when you do photosynthesis, that absorbs CO2. So obviously, when you're absorbing CO2, there's going to be less of it in the environment during the summer. But during the winter, when more cellular respiration is occurring within plants, they're releasing CO2. So obviously, CO2 concentration is going to increase. And the only reason this causes climate change is because our planet has something called the greenhouse effect, which means that when light from the sun comes into the earth, and it basically comes into the earth, it obviously escapes through radiating through the earth. But we have what's called this greenhouse gas effect, where all these gases in the atmosphere, such as CO2, nitrogen, and oxygen, I believe, all basically withhold that heat within. So if we know that carbon dioxide helps absorb some of the heat in the atmosphere so it can't radiate, and we're increasing that carbon dioxide concentration, there's going to be more CO2 in the atmosphere to absorb more heat. So that's going to cause, obviously, global, I don't even know why I say quote, global warming. So temperatures are going to get hotter. There's going to be more melting, rising oceans, etc. That's basically the entire idea with climate change is CO2 is rising, more heat is being absorbed, concentration of CO2 is increasing, more heat's being absorbed, it gets warmer outside, temperature increases, ice caps melt, ocean rises, blah, blah, blah. And once again, that can be harmful for a lot of species that, say, live near a beach or an aquatic ecosystem if water rises, for example. Our good old nitrogen cycle. Um, so another thing that's interesting is our environment is actually 80% nitrogen. The air around us is 80% nitrogen in two, but it's actually useful. It's actually useless to us because if you take chemistry, you understand how nitrogen into gas has a triple bond in between it, just with how the electrons work. It has a triple bond, and triple bonds are really, really hard to break. So we can't actually use any of the nitrogen that's in the atmosphere. So this process called the nitrogen cycle that happens in the ground mainly allows basically plants to grow because plants absorb this nitrogen and that allows them to grow. So we have different processes of the nitrogen cycle. We have fixation, immobilization, uptake, mineralization, also known as ammonification, nitrification, denitrification, leaching, and voltization. This is just a flow of how nitrogen goes through the atmosphere. So first we have N2, and nitrogen fixation is when N2 comes into the ground and it is converted into ammonia N3, NH3, by microbes. Microbes are doing most of this work. So microorganisms, basically bacteria in the soil, is doing this work, this conversion. So nitrogen fixation into gas comes into the ground. It is converted into ammonia. This ammonia is available for use and it is uptaken into the plants and it is used for energy. And nitrification is a process where, let me make sure I'm reading this right. I mean, you obviously can see it right here. You can read it. But nitrification is when ammonium, NH4 plus, is converted into nitrates by microbes. So nitrates and nitriums, NO3 and NO2, this is nitrification, is converted from ammonium. And once again, those nitrates are also able to be absorbed by the plants to grow. And once again, we have mineralization, also known as ammonification, is say when something dies or a human dies or an animal dies, or say a cow poops, and that goes into the atmosphere. All of those dead organisms basically are being broken down by decomposers, and that is going into the ground and being absorbed into the ground as nitrogen. So that's why, say, farmers would use manure to put on plants, say, hundreds of years ago, because that manure is getting broken down and absorbed into the atmosphere as nitrogen converted into ammonium, converted into nitrates, and then reuptaken by the plants so the plants can use it as energy to grow. And denitrification is the process of NH3 or ammonia that's in the soil that's not part of any plant. It's just out there in the soil. Is just basically reabsorbed back into N2. So it's just the process of it going back from ammonia into the atmosphere. So basically the opposite of fixation is denitrification. And then leaching would be, say, if it rained really, really hard and then nitrogen just basically got moved out of the soil. So this is actually a problem. So say a farmer puts a lot of, say, fertilizer on plants, leaching occurs, there's a big rain, a big thunderstorm, and that gets rained away, and all of that nitrogen gets, say, moved from the farm to, say, a lake or river or an ocean. Now we have excess nitrogen in the ocean or in a liver, or not the liver, a river. And that would be leaching, would be when water comes and basically washes those nitrates or ammoniums away. And then volatilization is another process where NH3, I don't know why I put that here, NH3 gas escapes back into the atmosphere. So basically the same thing is denitrification. So you can kind of think of denitrification and volatilization as the same thing, just basically how ammonia nitrates would leave and be converted back into nitrogen 2 gas. So that basically summarizes the nitrogen cycle. It's a lot of steps. You might want to look more into it yourself just so you understand how it works, but this is a very important part, probably the most in-depth part that you might need to understand. Dead zones. This is where I was going. So let's say this is why having a lot of excess fertilizer can be actually harmful. So say if a farmer uses all these artificial, say, sources of nitrogen and it gets washed into a river, right? I have a little diagram over here on the right. You might not be able to see it because of my face. But let's say, for example, all this nitrogen gets put on, all this excess nitrogen gets put on plants. It gets washed away into a source of water. That water absorbs all that nitrogen. That nitrogen goes down to the bottom of the water source, and it is absorbed by those plants down there. So say there's a lot of seaweed, a lot of algae in the ocean, that's going to absorb all that nitrogen, and then that's going to use that as energy. So all this seaweed or all this, whatever you want to call it down there, is going to grow, and those plants are going to absorb more O2, oxygen gas. They need it to grow. So we have all of these plants absorbing all this excess oxygen. So all these little fish and critter down there, they don't have any oxygen, so they're just going to die. And eventually it gets to a point where if you put so much excess nitrogen in an environment, it's going to basically just kill everything there and nothing's going to be able to grow because it's so hypoxic. Hypoxic means, hypo means low in general, and hypoxic, you can put together low oxygen. So when you put all this excess artificial nitrogen in an environment, that's going to cause basically the area to become hypoxic so nothing can grow and live there. And that's what's called a dead zone. So for an example from our notes is if you look through the left, we had a question and we had to put the different steps together. You can read through it, but basically the series is BDFCEA. So the question is, order the steps that result in low dissolved oxygen levels known as hypoxic water, meaning nothing can grow there. B would be fertilizer is spread on the land, excess fertilizer. That would be step 1. Step 2 would be D, nitrates and nitrites move into the streams. So step 1, excess fertilizer. Step 2, it gets absorbed into the streams. Step 3, that excess nitrogen promotes more algae growth in the water. Step 4 is C. Most of that algae goes uneaten or it dies or it blocks sunlight. Step 5, which will be E, is that algae is going to decompose and it's going to absorb all that oxygen in the process. And step A is going to be lack of oxygen is harmful to other organisms. So say if there's a fish that lives in the bottom of the ocean, Now they can't use any oxygen. Because even though fish, you know, I was going to say they breathe water, but they don't breathe water. They use their gills to take in water. They're still using oxygen from the water to survive. So even though they take in water, they still use oxygen. So when you have hypoxic water that is low on oxygen, those fish or those critters cannot live down there. It's like such a redneck saying critters. Oh well. Yeah, that's how a dead zone works. So that's why the idea of putting a lot of excess nitrogen or a lot of excess fertilizers in an area can actually become very, very harmful. And the biggest example of this is, say, the Gulf of New Mexico. So if you imagine, say, Texas, Oklahoma, Kansas, Nebraska, South Dakota, North Dakota, all those big farmlands in the United States where it's basically just flat prairie land where they just grow stuff. All of that excess nitrogen from all that farming area there, from all that fertilizer, all that nitrogen is getting washed away into that Gulf of New Mexico when it goes down south. So all that excess fertilizer gets poured into the Gulf of New Mexico, and then it basically becomes a dead zone and nothing can grow there. Another example is the Chesapeake Bay. In the northeast part of the United States, there's a lot of fertilizer, a lot of farming that happens in the northeast area. All of those little streams and rivers basically get filled out into the Chesapeake Bay in Maryland, and it becomes an environment where nothing can live there because the water becomes too hypoxic to basically support anything at all. Hadley cells. Hadley cells are interesting. It basically just describes how the biomes of the world work, and it's not by chance. If you look at, say, areas like the equator, if you look at areas around the equator, we have a lot of a lot of precipitation, a lot of rainforest, a lot of trees, a lot of rich diversity, say like the Amazon in South America or in Thailand. There's a lot of that part of Southeast Asia. There's a lot of, rich forest, a lot of precipitation. And this goes through cells. So at 30 degrees north and south of the equator, with how basically the air moves, we have a lot of low precipitation, a lot of high pressure air, and that creates a desert. So if you look at, say, Arizona, or you look at the southeast part of the United States, this will be considered part of a Hadley cell with 30 degrees north of the equator, where nothing really grows there. It's just a desert. And how this works is we have the equator. This is where, at the equator, is where the most amount of direct sunlight is hit basically on the earth. So the equator basically has constant hot air and heat coming in all the time year round. So what's going to happen is hot air rises and cold air sinks. So we have all this hot air hitting the equator, but that hot air is then going to rise. And when it rises, it then cools and condenses, and that causes rain. So that's why there's a lot of precipitation, which allows for a lot of growth, a lot of rainforest, a lot of biodiversity, a lot of life. But then as we move 30 degrees outside of the equator, say, for example, a lot of this cool air is going to get pushed out of the way, say 30 degrees north and 30 degrees south, and it's getting pushed out of the way because all this constant hot air is rising and all this constant hot air is coming down from the sun. It's going to push that cool air out the way to 30 degrees north and 30 degrees south of the equator. That cool air is going to condense and it's going to dry, and it's going to become really, really high in pressure, and there's going to be no precipitation. So it's going to be really, really hot. It's going to have no precipitation, and nothing's going to grow there. So if you look at this picture on the right, we have different cells of the earth. You have the Hadley cell, you have the Farrell cell, you have the polar cell. Another thing that's interesting is Antarctica in and of itself is basically just a desert because it gets no precipitation whatsoever. It's just a desert, basically. Just a cold desert. More like a tundra, basically. But that's how Hadley cells work. For my exam, what I'm going to do is I'm going to basically just memorize this, memorize the degrees, how the air moves, basically the entire process, everything I just described to you. That's what you need to know for a Hadley cell. Biomes. I just mentioned how biomes work. Obviously, biomes are a direct factor of temperature, so air and precipitation. And if you look at this cool little thing here I made on the left, it basically just shows you a graph. So if we have a really, really low annual temperature and no rain, we just get ice. But if we have really, really low temperature and little precipitation, we get tundra. So we just get Antarctica. Antarctica is tundra because it's really, really cold and there's no precipitation. And in an environment that, say, is really cold, but it gets a lot of rain, we get what's called a boreal forest. So think of areas like Alaska or Russia. That's a boreal forest, or parts of northern Canada. Um, and then once again, if it's really hot, but there's little precipitation, we get a desert. So say Southeast America, parts of the Middle East, it's really hot, no precipitation, we get a desert. And then if it's also really, really hot and there's a lot of precipitation, we get a tropical rainforest, which would be like, say, the Amazon or parts of Thailand, Southeast Asia. But North Carolina is actually a temperate forest, which I guess would fit it well. In North Carolina, it's not cold, doesn't get super hot, it's not a lot of rain, it's not dry either. So you get more of like a temperate forest, basically. So I think that's pretty interesting. If you look here on the right, it's basically a diagram of how all the different biomes are set up. Ideally, if everything were according to plan, it would look perfect. So going this entire way, everything will be one color. A little bit lower, everything will be one color. A little bit lower, everything will be one color. But the reason this doesn't work out is because there are a lot of geographical barriers. So say some areas are more mountainous than others. Some places have more wetland than others, right? So that's why everything isn't exactly perfect, if that makes sense. So I find that interesting. Another thing that you might want to understand, you would think that a lot of people would understand this, but it's just something that I guess I need to point out, is climate versus weather. So weather obviously is what is happening right now. So right now, where I'm at, it is September, what is it? 23rd. It was 80 degrees outside today. It wasn't raining. That'd be the weather. Climate would be the overall environment over time, right? So obviously, if you ask me a question of what is the average climate in North Carolina during January, it's not going to be 80 degrees every single day. Absolutely not. It's just not how climate works. So more predictable is climate, because climate allows us to follow long-term trends. A varied pattern is weather, because weather is obviously more variable. Less predictable is obviously weather. Climate sets long-term trends, and then weather follows those long-term trends. And obviously climate is more consistent. So during the climate, if we were looking at climate, we know just in general, summer is more hot than winter is. So in July, obviously we can try to predict the weather if it's going to be hot, but obviously we're going to have some days where it's colder and it's warmer than others. That's just the difference between weather and climate. You think that'll be common knowledge, but just might be something you might want to know for the exam. Another thing is how our seasons and how the tilt of the earth works, and what allows us to have seasons. So if you look here at the bottom left, this is basically an imaginary planet that has no tilt. It's not tilted like the earth is, and it's in a perfectly circular orbit. So the earth actually isn't in a perfectly circular orbit. It's more in like an oval shape. So the earth is more like an oval, but this imaginary planet, say, is in a perfect orbit and it has no tilt whatsoever. So if you look at the top left, basically the earth's tilt allows us to have seasons. So say where I live in America, um, and during the winter, the earth is tilted more away from the sun, at least in the northern hemisphere. So that's why it's colder. but during the summer, the Earth in the northern hemisphere is tilted more towards the sun. So we get more direct sunlight in America, and that's why it's warmer in July than it is in January because of that tilt. Also, um, if you look at the top right, what I basically just mentioned to you earlier with Hadley cells is the equator gets a lot of direct sunlight, where the closer and closer you get to the poles, get little direct sunlight. That's why it's so cold in Antarctica and at the North Pole and like Greenland, basically all year long, because it gets no direct sunlight whatsoever or very little. Um, yeah, another thing I wanted to mention is if we have a round orbit, all the exact day lengths are going to be the exact same. So you know how in America we have like daylight savings, where basically our days get shorter during the winter and during the summer they get longer, and that has to do with how the orbit works. And say some days it'll be like, I know during the winter the sun comes up at what, like 6 in the morning, and it goes down at like 5 p.m. So the sun's out for say 11 hours during the day. But during the summer, the sun will come up at like 7 in the morning, and it won't go down until like 8 or 9 o'clock at night. So the reason during the summer we have longer daylight hours and during the winter we have shorter daylight hours is once again because of how Earth's orbit and tilt works. So if we had an imaginary planet that had no tilt, it, and it had a perfect circular orbit, literally there would be no seasons. We would have 12-hour days, 12-hour nights. Nothing would vary whatsoever. There would be no weather changes anywhere. We would have no north and south poles, no equator, because every area gets the exact same amount of sunlight. But that's one thing that's so interesting and beautiful about Earth is with the way it's like perfectly tilted and with the way the orbit works, which is what allows us to have these different seasons. And then obviously we have photosynthesis and cellular respiration and those different processes that give us fall and give us spring and whatnot. So really cool. All right, so this is a practice exam. My class gave me a practice exam just to go through to help you with your studying. It does help you because just like with many things in life, you can read and talk about something all day, but until you say actually practice it, you don't you're not really going to understand the material in full until you practice it. So just looking at a question like this is over 30% of the earthworms we find in North America were either introduced, blah, blah, blah. What's the question? Which of the following ecosystem services have the worms most directly impacted? So if you actually read through the question, earthworms are detritivores. I don't even think I said that right, which means they depend on dead organic matter to acquire their nutrients. So this has to go with what I just went through. If you go to the very beginning of this video where I talked about ecosystem services, supporting ecosystem services basically have to do with nutrient cycling and soil processes. So the answer to this question would be supporting ecosystem services. Another thing is, if you look over here on the left, we have more practice questions. You can pause the video and read the question, but I'm not going to do all that because you can read it. So just go ahead and pause it, read it. I'll explain it. So the question here is, what's the species richness of this area? It would be 6 because if you read through it, we have 6 different types of species. Over here on the bottom left, what was the question? What other kinds of biodiversity did Grace document? There are different multiple answers. You could say genetic diversity, functional diversity, ecosystem diversity, because you basically took functional diversity, what's it doing, um, ecosystem diversity, because where it's found. A lot of these answers are very subjective. I'm going to be completely honest. A lot of college exams are very, very subjective. So I can't necessarily explain it as well, but go through a practice exam. It'll help you out a lot. Another question at the bottom left here is when Grace collected her data, she first randomly selected 6 locations on her property and then walked between those locations. Did Grace sampling's technique include replication within the acre plot? Yes, and she did use replication because she basically just did the same thing multiple times. She just walked in straight lines, And we just described earlier that walking in a straight line is an example of transect sampling. So yes, there is evidence of replication using transect sampling. At the very top, we have a question of, after sampling 150 individual beetles, which site had the lowest species richness? And it would be the no traffic, because as you can see using the refraction plot, no traffic has the lowest number of species with the same amount of sampling effort, if that makes sense. And then I'm assuming with these bottom questions you can't see with my face. And this is, this goes back to the Shannon Wiener index, or Wiener index, however you want to say it. We have our question here on the left, which is basically the number of individuals, the total number of individuals, and it tells you on the left here the correct Shannon index. So for the first one, for Huggins Island, the correct Shannon index is 1.41 to 1.43. And for Bear Island, the correct Shannon index would be 1.13 to 1.15, and it gives you all the number of individuals there. And on the right here, I've written down some math with basically for, say, Bear Island, I put the top, I did log of 8 divided by 93 multiplied by 3.22, multiply again by the fraction, plus log of 22 divided by 93 multiplied by 3.22, multiplied by the fraction again, plus log of 63 divided by 93 times 3.22, multiplied by that fraction once again, and we take the sum of all of that, and that gives us 1.14. So if you can see that math, it literally lines up exactly to what the correct answer is for the two questions right here. And if you go back to this first slide right here, it basically just explains everything, and it's the exact same correct answer. Dang, I put a lot of test questions in here. So 7 at the top. A local real estate developer would like to build a hotel with affordable housing. If the developer wants to make a decision based on the Shannon index variable is H apostrophe, which of the following should they do? And the correct answer would be they should choose an island with the lower Shannon index value. So if we want to build an apartment complex, we would want to do that in an area that has a lower level of biodiversity. Because like I mentioned earlier, if an ecosystem already has a low level of biodiversity, that means that there's probably a lot of already like dead soil there anyway. So why are we going to build an apartment complex over an ecosystem that has a lot of rich soil, a lot of life, a lot of biodiversity? So if you get a question saying there's two areas, one has a really low Shannon index value, one has a really high Shannon index value, which one would you want to build homes over? You'd obviously want to build one over the area with less biodiversity. So that would be the lower H value. All right, 8. If a species of plants were introduced to both Huggins and Bear Island, what would this do to the alpha diversity for both islands? Once again, alpha diversity is just the number of species in the environment. So if you just added a species of plants, that would just increase the alpha diversity of each environment by 1. Number 9 is if the species of plants were instead introduced to only Huggins but not Bear Island, how would this impact the beta diversity for two islands in the short term? Remember, beta diversity has to do with the two species that the groups do not share. So if we were to add both species of plants to both islands, the beta diversity would not change while the alpha diversity would. But if we're only adding the species of plants to one island, then it's not shared among the other islands. So when we look at the beta diversity between the two groups, it's also just going to go up by 1. And over here on the right, I don't even know if you can read those questions because I know my face is in the way. I can go over the top 2. The first one, again, we already did that question. Number 11, if you can see that, the researchers are confident that they exhaustively found and identified all possible beetle species at their research sites. Do the graphs above support this assertion? And the correct answer is no. All 3 treatments show that new species continue to be identified with additional samples taken. Because what we said earlier, if there's not an asymptote, that means they're still discovering new species. So when we look at that refraction plot, and it's still growing kind of linear. I mean, this one isn't really linear. It's kind of decreasing, but it's not at an asymptote yet. It's still increasing. So that's why how we would know that, no, all 3 treatments show that there are new species that continue to be found. All right, 14. In the Midwestern United States, prairie ecosystem, an individual grass plant captures X units, blah, blah, blah. If 858 units of solar energy is measured, what is that? Okay, so the question is basically asking us to calculate GBP, gross, because it's telling us how much is used in respiration, and only 859 of it is available, which will be net PPP or in PPP. So if we want to calculate the GPP, which is what it's asking it for, which is what it's asking us, we just add respiration to net primary production, and that gives us GPP. So when you add them together, X would be 1006, which would be GPP, which would be the original amount of energy in the ecosystem. So 1006 units, whatever. 15, you can read through that. Basically, all the answers are correct. 16, you want to measure the respiration of your favorite houseplant. To do this, you first place it in a clear chamber. Then what do? Once again, I talked about this earlier. The correct answer is measure the amount of CO2 produced at nighttime, then use this to do your calculation. Because like I said earlier, plants do most of their respiration at night. That's just something you're going to have to remember. 17, several factors are contributing to the measurable. My dad calling me. Sorry about that. My dad called me. Sorry about that. 17. Several factors are contributing to measurable rapid sea level rise on Earth. The first is thermal expansion, which increases water volume. Which of the following has been shown to be a key factor? Yeah, obviously additional melting, because as there's more climate change, more CO2, there's going to be more sea level, thermal expansion, more water volume, and then obviously there's going to be more water melting from snow and ice. So I'm trying to look at some of the other answers to see if any of those make sense. So E doesn't make sense. Lower evaporations over the largest oceans. I don't know. Additional water from rain over land, that doesn't really make sense. Increased rain over ocean, that doesn't really make sense. So yeah, 17 is the only thing that really makes sense, because the question is asking which of the following has also been shown to be a key factor to increasing water volume. And obviously, additional water from melting snow is obviously going to create more liquid water. And number 7, local real estate developer. I already talked about this one. You would obviously want to choose the area with less biodiversity if you were building houses or apartments, right? And then yeah, I think I just copied and pasted the same slide again. I must have messed it up. So yeah, alpha diversity would increase if you add species to just 2 groups. Yeah, makes sense. Another thing is what I mentioned earlier. With this test, you can. This is what I've learned with school in general. You can review notes. You can read the textbook all you want, but until you do actual content practice and you practice the question and you actually think about it, because I've taken tests before where I've thought I've studied really, really hard, and I get the test and I have no idea what I was doing, because you have to do like application-based questions to like show your knowledge, right? So personally, what I've done is just go into ChatGPT, send it a PDF of your learning objectives, and then also send it, say, a list of notes or say a practice exam, and ask it. Say, hey, this is my practice exam. This is my learning objectives. Generate me a list of 100 practice questions for my exam. and just go through all those different practice questions. So earlier today, I asked ChatGPT, and it was like, hello, below, there are 100 practice questions with answer choices provided. This gives you a comprehensive set, and basically just go through all the questions and answer them, and then just ask ChatGPT, give me the correct answers, and you can grade yourself, and you can learn from each question. So that's what I did. So I did a practice question earlier today with 100 questions. I got 91 of them right. So I did 100 questions from ChatGPT, and I got 91 of them right. So that's just my personal advice to you. You don't have to follow it, but if you really want to do good with any exam in general, or especially an exam for this test, go ask some AI. Be like, hey, generate me some practice questions. Because if you do one practice test, those questions are going to get old fast. After you do the exam one time, you're going to learn it. So you need more content. So, I mean, AI is crazy nowadays. So, yeah, use it to help you out. Do some practice questions. That concludes my review. I think that's all the slides. Like I said, I want to do this primarily for my own studying in general, because I obviously have to take a test on this in general. But I mean, I figure, say, if I leave this video on the internet for, say, 10 years, and then 1,000 students that take this same course rewatch my video later and they find it helpful, then there's no reason for me not to make this video. So I really hope me going over all of this definitely helped you. I know it's definitely helped my studying, having to go over all this and making sure I understand everything and all this different vocabulary and all this math. So if you found this video helpful, good for you. I love each and every single one of you very much. Thank you for watching this video and supporting me. Goodbye. Good luck on all your exams. And goodbye, my Parker Knights. Have a great day. I love you. Goodbye.